Hey, so I'm going to try and help you to review some of your information from Unit 6 so that you can then better be capable of answering some review questions. Okay, so just follow along and we will do our best to make sure that we feel like we know what we're doing. So, first and foremost, we talked about in unit or topic one RNA and DNA, right? And the big key thing is really that they're both used for hereditary information, and they both have nucleotides that are their primary monomer that is used in the conversation of moving from one um, thing to the next, right? Um, and so we can look at gene expression question, what's it mean, or what do you think it means when scientists say the genetic code is universal to all organisms, right? This is basically that we all have RNA and DNA. We all have these nucleotides, and those nucleotides can be used to create a protein, and that's really important. It's a common language, right? Um, so, what evidence do we have that all organisms have common ancestry? They have DNA, right? And you can look at this picture here and you can see how there are A's paired with T's, right? So if I have 100%, if I had 30% of A's, right, in my DNA, then it would mean I'd also have to have 30% T's, right? So a total of 60%. And then the other 40% would be G's and C's, okay? Because they pair up, you have to have about the same percentage for each, okay? Um, when we look at them, purines are the ones that are G's and A's. They have that double ring structure. Pyrimidines have a single ring structure, okay, over here. Okay. Um, the pairing rules. Trying to think what else in DNA. When it comes to them being translated, not all transcribed or translated, right? Not all DNA is important. Sometimes we have what are called introns, which are those regions in eukaryotes, especially, that are not actually going to be used for transcription and translation, right? But prokaryotes are small, so they generally don't have these introns. Our DNA is huge. It gets wrapped around little histones. That can be part of gene expression, because if they're tightly coiled, they're not going to be expressed. If they are, not tightly coiled, or if they are tightly coiled, they won't be expressed. Okay. Let's see what else. You already know DNA is double, RNA single stranded. Stability is a big key thing, right? You want to have that stability to your strand, which is why you have a double strand for DNA. It's more stable. RNA is not as stable because it's single stranded. Okay. When it comes to transcription, right, you're always going to take the template strand, which goes 3 to 5, and you're going to make your mRNA from 5 to 3. And just remember that, the difference is that if DNA has an A on it, you're going to end up transcribing a U, right? And when we talk about replication, right, DNA replication, this is kind of similar to this process of transcription, in the sense that you're going to make your what's called your leading strand, right? You're going to go from a 5' to 3' area. So this RNA polymerase binds right here in the center, and then it moves this direction. Okay. And the Okazaki fragments are actually going to go 3 to 5, and they go in little snippets. Okay. Let's see. I'm just going through the Unit 6 presentation to try to help with the different topics, right? You already know about the central dogma, right? But DNA gets transcribed to RNA, and it's the same language. And then RNA gets moved into being read by codons, right? You read those 3 codons, and then you can create the protein from it. And so there's a specific amino acids that code for that are coded for by different 3 nucleotide sequences, right? You have 3 different types of RNA. You have transfer RNA, messenger RNA, ribosomal RNA. Those aren't the only ones, but those are the primary ones involved in gene expression, right? And so the tRNA here, you can see, are attached to the amino acids, and they're going to have what are called anticodons that are opposite of whatever the mRNA codon is, okay? And then the ribosomal RNA is actually what binds the ribosomal proteins to form the ribosome. So it's kind of like the RNA is the rRNA is the ribosome in a sense, okay? You have 3 steps to transcription: initiation, elongation, and then termination, right? And it's all about that promoter, right? You got that promoter on your DNA that attracts the RNA polymerase. Then once the RNA polymerase has made your mRNA, it's going to get to a place where it's told stop, right? You get that stop codon, and it stops to from continuing to build, right? Let's see. Prokaryotic transcription occurs in the cytoplasm, occurs really quickly, literally happens in the same step because it's in the same place. Whereas in eukaryotes, it's much different, right? The transcription is actually in the nucleus, then it goes through nuclear pores and ends up leaving or exiting at the end, right? Okay. In eukaryotes, you have this post-transcriptional process where you have your pre-mRNA getting cut into little sections, which allows for, once you remove those introns, those exons to be put together and form different variations of the same mRNA strand. And so you can actually then lead or increase the number of proteins that might be created. Okay. And if you look at this diagram here, there's a bunch of things to fill in, right? But essentially, I don't have those all right here. You can go back to this picture and you can see where, each of those letters coordinates with the things on the previous screen. And here's what I was talking about with alternative splicing. Okay. Let's see what else. In 6.4, we talk about translation, right? And again, initiation, elongation, termination, this whole idea again. And the big key here is using the codon chart to identify what are the amino acids that I hit, right? When I get to a UAG, it's a stop codon, right? AUG is always methionine, but it's my start codon, okay? So you can have a methionine in the middle of a sequence, but if you've not hit one already, it's the start codon. So that's kind of like where the RNA polymerase knows, let's start making my, mRNA strand, my protein, right? Let's see what else. Topic 6.5, we're looking at the regulation of it. Again, big key here is with the difference between operons and just a regulator gene in a eukaryote. So operons are what we call that stretch of DNA that specifically codes for a specific protein to be made in prokaryotes, right? So you got an enhancer region, which is kind of like your promoter. Well, I shouldn't say it's a promoter. The enhancer region is actually only in eukaryotes. So this right here, I apologize. This right here, this is an example of eukaryotic gene regulation because you got an enhancer region and then you got your promoter region. Okay. See, find another important piece of information. Sometimes prokaryotes and eukaryotes have genes that are being coordinatedly regulated right at the same time. Okay, that's important. So as one is being regulated, it's causing another one to also be regulated. You can see here where we have the prokaryotic coordinated regulated gene expression within the lac operon, which is inducible. So inducible meaning that it's got to have something that attaches to start the transcription process and then essentially make the proteins. Whereas if something's repressible, like the trip operon, it actually has to have something that stops it, that attaches and stops it at the operator. Okay. We move down to these regulatory switches in eukaryotes, right? also called enhancers. They connect to the mediators. These are these things right here, right? And those have to be there to make the gene be transcribed, okay, in eukaryotes. And so some cells don't have these regulatory switches get activated by the proteins that activate them, or have these things here, these transcription factor complex proteins, right? These general transcription factors or mediators might not be there, and so you might not have transcription or translation occur, right? And then I'm going to move to the next topic. Or did I skip 6? Okay. I've kind of already talked about these things. And so we'll look at 6.7, which is mutations, right? So there's a bunch of different mutation types. The big key is that you have those two categories, right? You have your frame shift mutations, right? And then you have your point mutations, right? So point mutations are often called substitutions because you're just moving one nucleotide. So they might not have a big effect. In fact, they might sometimes not even if it's especially the last nucleotide in a codon, right? But if I have a frame shift, where's the frame shift example I wanted? If I have a frame shift, right, that's like a deletion. So something's been, or an insertion. Those things are going to cause the whole codon sequence from that point to be moved, right? Now, what would happen if it was in an intron? Well, because introns don't code for anything, it really actually wouldn't make a difference, right? But if it happens in a regulatory sequence, right, that could make a big change and cause a new protein, or even the protein to not be created as well as it should be, right? And then when it comes to biotechnology, one of the big things with biotechnology, which I don't have on here, but just, you have things like PCR that we just did, where you're duplicating and replicating your DNA so you can get a bigger picture, and then you cut a section of it, as well as you have your gel electrophoresis, which can be used to move the DNA, and then you can analyze the DNA and compare it to other strands. And so those are all important pieces to know and to just be aware that we are changing DNA or making it what we want it to be all the time, right? And there's plenty of other things I could go over right now. I just don't want to have you spend way too much time, going through this. I would say you could look through this PowerPoint if you need some help. Another key place to look or to go over would be to review the course exam description for Unit 6, which you should have access to on Google Classroom. And it'll give you a lot of the key pieces of information. So I hope that helps, and I hope to see you complete the assignment on AP Classroom that has questions for you to review and to hopefully do better on than you did on your assessment, and I can give you some points to increase your grade on the test. Let me know if you have questions.